Okay. So we've kind of gone over the basic, how to use all this, these software platforms, but what I really haven't covered is what are the kind of results that you give your collaborator. That, at some point maybe you might not be doing your own project, but obviously doing somebody else's. And some of you actually are, I've given you projects to actually do for other people. So what I'm going to do today briefly, and I won't take too long on this, is basically show you what kind of things what you need to put together in order to give somebody a bioinformatics report, okay? So what I'm going to do is I'm going to start with IPA first, and then I know we don't, Partech, the license isn't working right now, but what I'll do is I'll use a report that I already sent somebody else to just kind of give you an example of some of the graphs and some of the metrics that you'd want to include to describe somebody's study, okay? Can everybody open up your IPA? Or does everybody have their IPA open? Okay. So as an example, what I thought I would do is go back to our stem cell study that we did as an example. And when we were going through the different lists, so basically what I've done is under our projects, I've selected, and it should be day and cell type 2A ANOVA. These were the top 1,000 genes that were significant due to both the type of stem cell it was and whether it was differentiated or non-differentiated. Okay? So you can just double click this, and I've already opened mine up. You double click it, you should get this screen here. This is what we're going to use as our template. This is what we're going to download. Obviously not everybody has a copy of IPA. So what you're going to need to do is basically take IPA's information and download it into an Excel file that you can then give to somebody. Okay? So the first thing I do, in the first tab in my Excel spreadsheet that I'm going to give to somebody is going to be all the genes that were differentially expressed. So what I do is I go this farthest tab here under molecules. Okay? Click that. I know it says molecules, but it's actually our genes. And basically here you just get a list of not only our genes that were in our study, or at least in our gene list, but also chemicals and other genes that are associated with some of the networks that the program created based on our gene list. Okay, so here's what I want to do is I want to go here and I want to say export data. Okay. And usually what I do is, it'll go to data export settings. Usually I just do it in Excel. Maybe. There it is. Okay, and then hit OK. And I'll just put this on my desktop. Oh, I gotta name it something. Test 2. I don't know. Hit save. A lot of times on some of your programs, the Excel file will open up automatically. It's not doing that for me. So what I'm going to do is go to where I saved it, my desktop. There it is. And open it. So your file should look like this. Can everybody see that? Is that a little better? Okay. You can see this, any platform you use, they always want to put their name on everything. Yeah, I love the fact that it's Kaijin, but guess what? This top line really messes up my sorts. So what I do is I just select this top line and hit delete. Now what you're going to see, and again, I'm recording this, so if you get lost or something, don't worry about it. You can always go back to the video. Usually I'll bold this. Now here's the thing, is most people that I work with, they're not going to understand why, 2,5-deoxydribenzoic acid is on their gene list. So my first thing is to just give them the genes that they're familiar with, okay? So in order to do that, I got to get rid of everything else. Any ideas how I would do that? Yeah, I mean, you could do that too. No, not location and type. What did you notice about these things here that weren't on our original gene list? They don't have any values in the p-value and fold change, right? Yeah. Honestly, half of bioinformatics is sorting. It really is. Like when in doubt, sort. So you can go to fold change and just sort by, I don't know, descending or ascending. There we go. So what we can now do is we can go down to wherever the fold change information stops. And basically select, so this is, the devil's row, 666. Go to 666. Go all the way down. And just get rid of all this stuff. You know, if it was for me, I'd probably leave it because I like to see what networks some of these things are in. But honestly, for a collaborator, that's just going to confuse them. And the one thing in bioinformatics you do not want to do is confuse your collaborator because it makes your life miserable. Oh, and then while I'm at the bottom, here's something you can do, and I highly recommend this, is to basically color your p-values. Making data visually appealing, and I know this is kind of contrary to, bioengineering in general and computer, everybody's just used to like looking at numbers. For most people though, a color, just adding a color is going to improve everything. So here's what I do is I select my fold change, go all the way up. I should have shifted. Oh well. Okay, right now, this thing is reading this as text. So what I want to do is, so on my Mac, this thing will come up, and you can do this any way you want. You can repaste this, format it. You just have to convert this to a number. Okay? Then you want to hit your conditional formatting. And this might be review. A lot of you might be like, of course we know how to conditional format, but you'd be surprised how many people don't. So I'm just going to show you anyway. Even if you don't know? No. Okay, well you're going to learn now. So here's what you do. So I can make any rule I want. So I'm going to go new rule. Aha. Now here's what I want to do. Is I don't want the 2-color scale. I'm going to go for the 3-color. Okay. Usually my lowest values, and this is, you see this in IPA. I usually follow IPA's format. Honestly, you can make these colors anything you want. You can make them plaid if you really wanted to. And there is a way to do that. I've had my high school students did that. I was like, how'd you do that? So the lowest values, we always want kind of that green color. Let me see if I can find it. There it is. Spring green. Remember, when you're looking at IPA always gives the lowest values in green color. So I'm keeping with that. We don't want the 50 percentile. I want the number. So I'm going to put a number and I'm going to put 0. Because remember, this is fold change, so anything negative is going to be green. And I'm going to make this, let's make it white. Now this highest value, I'm going to make red. Okay, and I hit OK. Now we have it color-coded. Sometimes you might have to play with a scale. Some of these genes, like you might get a study where most of the genes are like -1, -2, somewhere in that range. And then you might have one that's like -50. It's going to throw everything off. So what you can do is you can adjust that scale. Instead of going highest to lowest, you can always go, say I want my highest number to be +10 and my lowest -10 or something like that. But I think this adds a lot. And you could even, I'll do this with the fold change or p-value sometimes too. I mean, all of these genes are significant. But if I had all my fold changes on here, what I might do is just highlight all the p-values that are below, that are significant, like bright yellow. So you can see how this, this is working here, is that all I have to do is kind of look at the list and get a feel of, what these genes are doing. I can sort by, the symbol here. And you can see, really clear now what genes are going up, what genes are going down. I highly recommend formatting your Excel spreadsheets that you give to people. Okay, and usually I'll just name this sheet like genes or something. So this is our first tab. This is what the first thing you want the people that you've analyzed their data for to see. Here are the genes that are different in your study, right? And a lot of people won't even make it past this tab. They'll just sort by fold change and go, oh, there's my gene. Okay, go back here. Where's my IPA? There it is.